All right, everybody. Welcome to your first assignment for week number 4, which is going to be talking about protein folding. This is kind of a continuation of what we learned in Unit 7. So if you want to know the sequence of this learning, in Unit 7, we learned about DNA to RNA, RNA to protein through transcription, translation. Then we took the quiz on this first week of digital learning. And then the last 2 weeks, we kind of put that on pause. And we learned about viruses and the immune system. And last week you learned about how to protect your body, how to keep yourself safe, made a procedure, how to go to the grocery store. Okay, and now we're coming back to that DNA level of understanding. So this lesson is going to get into, so DNA codes for the RNA, RNA codes for the protein. Then what? What does the protein do? What happens? So the protein has to fold in order to become what it's supposed to be and be able to do what it's supposed to do. So if that protein helps you digest food, the amino acids need to be made, and then it folds itself up and then goes and digests food. If it's a protein that's designed to make your nervous system function, then the protein gets made in the amino acids, and then it folds itself up, and then it does what it does. So until it folds itself up, the protein doesn't do what it's supposed to do. And you can actually unfold proteins and keep them from doing what they're supposed to be doing in some more advanced biology techniques in labs and things like that. It's called denaturing the protein. You actually unfold it. So the protein not only needs to be made with the amino acids in the correct order, but it needs to fold in its proper shape. And then that folded protein then travels around the body and does what it's supposed to do. So what we're going to talk about are the 4 stages of that folding here in this lesson. So let's jump back into some learning. We'll talk about it in phrases, some notes. All you're going to need for this is some paper if you want to write this stuff down, and a pen. Nothing fancy on top of that this time. Perfect. Let's focus that thing in. Okay. So where do we start? Protein folding. Okay, and I would be remiss if we don't start what we did in Unit 7, good old central dogma of biology, which was our DNA code contains, the DNA holds the information, which is coded into an mRNA or a messenger RNA, which then takes that message out to the cytoplasm and makes our protein. Just remember that a protein is just a bunch of amino acids together. I'll show you that again. And then let's do a little quiz check right here. DNA turning into RNA. What's that process where it reads the DNA and it makes RNA? Do you remember? Yeah, it starts with a T. Which one? Transcription. And then the one where it reads the RNA in the ribosome and that makes the amino acids coming out the top? Yeah, starts with a T. Translation. Okay, we went over that series of steps in Unit 7, so hopefully you're feeling like you remember that. So what we're going to be talking about is this step right here. Once the amino acids get made, how does it fold? Okay, so at the end of translation, you have amino acids together in a certain order. Okay, so remember I'm just going to put AA for amino acid connected to another amino acid, connected to another amino acid, connected to another amino acid, connected to another amino acid. And we got these from that genetic code, which we will go over in just a second again. And I'm just going to draw a couple of these. These can actually get hundreds and hundreds of amino acids long. Don't have the paper, the space, or in this case, the patience to do that. But just so you know, amino acids in order together, this whole thing is called a protein. It's kind of like beads on a necklace or pearls on a necklace. And each pearl is an amino acid, but the whole necklace is called a protein, if you want to think of it that way. Okay, So this thing has to fold up. It has to get all folded on itself. So it like bends and twists and turns. And then what are those different levels of folding? So the different levels of folding. Levels of protein folding. Okay, the first level is called primary. And we use this little degree symbol for 1 to the degree power. That's kind of the symbol for primary. So the primary step or the primary level. What happens at this level? All right, basically this is the primary level. It is just the amino acids in a long chain. Right here. So I'll even point that. Okay, that is your primary. When it comes out of the ribosome during translation, it is already coming out in primary. All right, I'm going to redraw that right here as just kind of like a line like that. That's primary. So instead of me drawing all the little beads, I'm going to just draw a line to represent the chain of amino acids. So this and this are the same thing, because now you're like, where did all the amino acids go? They're on there. I'm just drawing it as a line because, you know, as scientists, we're lazy people and we like to keep it simple. Okay, so primary is just the chain of amino acids. Okay, next you get secondary, which now 2 with a little circle. And what happens at secondary? You get some simple folding in different structures or different shapes. Let's do different shapes. And there's basically 2 different shapes that it folds in when it first starts to fold up. You get something called an alpha helix, and you get something called a beta sheet. Okay, so I'm going to take this long string right here, and what's going to happen is it's going to start to corkscrew a little bit. So it starts to come and it kind of corkscrews around itself. That's an alpha helix. Okay. Remember, DNA is a double helix. It's 2 of these because it's got 2 sides of DNA. Does that kind of ring a bell? Well, protein doesn't have 2 sides. It just has 1 string. So it kind of spirals around. That's an alpha helix. Okay, so that's some basic folding that starts to happen during the secondary step. What is a beta sheet? Beta sheet is kind of a zigzag. So it kind of goes like this, and it kind of goes zigzag, 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 and then it'll kind of fold, and then it'll come back and go zigzag, zigzag, zig, and then it'll go like that. So that area right there is called a beta sheet. So if you get a little corkscrew action, that's called an alpha helix. A little zigzag motion, that's beta sheet. So during the secondary step, you get some alpha helix forming and some beta sheets. You could have both of them. Maybe it's an alpha helix here, then a beta sheet, then an alpha helix, then a beta sheet. So you can have both of these happening. It's just that when it forms that shape, we call alpha helix. When it forms that kind of shape, we call it beta sheet. Okay, that's secondary step. The third one, called tertiary, which is 3 with a little circle. All right. Honestly, at tertiary, it just starts folding all over the damn place. Okay, it starts going all ziggy-zaggy, weedy-wacky. You can't even predict it. This is where proteins just kind of do their thing, and you don't have much control of it. So the protein folds all over itself, in different directions. Just all over the place. And so you go, well, how do you draw that? How do I draw that? I go, or maybe a little alpha helix right in there, or, you know, beta sheet right in there, something like that. And you go, damn, that looks like a globular mess. Yes, this is what we call globular, which is another way of saying it looks like a glob. It looks like just a bunch of stuff all folded on itself. Yeah. So that's kind of what it looks like. It's all folded up and twisted around, kind of like your earbuds stuck in your pocket. They just kind of go all over the place and twist in your pocket. That is tertiary folding right there. Okay? And there is one final step, which is called quaternary. Oh, that's a fun word to say, quaternary. It's another one of those words to screw around your parents with. Quaternary. Like quarter, quarters, 4 quarters. Fourth step. And so this is a finished protein. This is a protein right here. It's done. So you're like, well, but there's one more step. Okay, this is where different proteins combine with each other. To make a super protein. It's not really a word. I'm just kind of making it up. Okay, you guys used to watch Power Rangers when you were young. You know how like everybody had their own little like animal, and then all the animals would combine to make the Megazord. Okay, well this is kind of the same thing. I've got a protein right there, so let's call that protein A, and then I've got another protein. Call that protein B, and these two come together and make, oh, how am I going to recreate this? A super protein, which is protein A and B combined. And that's quaternary step. So how does it start off? Out of translation, we get an amino acid sequence, which is a nice line. We just draw it as a line. You start to get some basic alpha helix formings or beta sheets, and then it goes all wiggity wackety, folds all over itself. And then when it's done folding all over itself, one protein that folded all over itself combined with another protein that folded all over itself make a super protein that are combined. And sometimes it's even more than 2. It could be 3, 4, sometimes even 5. And that is your 4 steps of protein folding. Primary, secondary, tertiary, and quaternary. And that is protein folding. So let's jump over to your online notebook and say, okay, what am I supposed to do with this information this week, Mr. Charlton? Good question. So here's week 4. Go down here to assignment number 1, protein folding. This video is the first one, Mr. Charlton's video on how proteins fold. There's a couple other videos that you might want to check out here just to learn with better pictures than what I'm drawing, but I gave you the basics. And then I say this, okay, so on a separate sheet of paper, I'm going to give you a DNA sequence. So you copy this down as your DNA. Then you're going to transcribe that into RNA. So remember, when you make RNA, what goes with a T? It would be, I'll even do it with you right here. Oh, the fun of watching the videos. You get little cheats. I'm going to make it the same letters as this. Here we go. A goes with T. What goes with A, ladies and gentlemen? Well, T, Mr. Charlton. But there is no T in RNA. So there's a U. What goes with C? G. What goes with A? T. Goes with A? T again. What goes with C? G. What goes with G? C. What goes with T? A. What goes with G? C. What goes with C? G. What goes with A? U. What goes with G? C. And I'm going to let you finish the rest on your, you didn't think I was going to give you all the answers, did you? Go back. Okay, so that is our mRNA. And that's what I ask you to do here. Transcribe the DNA into mRNA. Step 4, translate the mRNA into amino acid sequence. So you go, wait a second, amino acid, how am I supposed to do that? You remember our little chart right here? Remember this guy? Woohoo! I put it here. It should also be glued into your notebook if you have your notebook available. And you remember how to use it. Start at the center and work your way out. So I go, all right, A, U, G. I want to make this even bigger. So let's take a look at it. Come on, baby. Come on. Maybe I shouldn't mess with it. Some bad things happen. Okay, let's start here. AUG. AUG is methionine. So I'd go up here and I would put met. TTG. I go back down here. Wait, sorry. Did I put TTG? Oh my God. Didn't even catch my own error. I said it, but I still didn't follow it. Hopefully you caught that. UUG, my mistake. UUG. All right, UUG, leucine. So go right here, put leu. CAC. CAC is histidine or his. GUC, go down here. GUC is valine or val. Oops. And you will finish that off as you finish off the rest of it. Okay. All right, that's step 4. Step 5, it says redraw the amino acid sequence as a line. That's like what I just did with you at the beginning of this video. So if you don't know what to do in number 5, rewind this video and go back, watch the front of it. Then draw some secondary folding, which is alpha helix and beta sheet. You can draw them any way you want, how you want. It's totally up to you. And then draw it as a tertiary. I said you do not need to make quaternary, so don't worry about quaternary. So what am I going to see on your paper? I'm going to see a DNA sequence, an mRNA sequence, an amino acid sequence, a primary, a secondary, and a tertiary. Okay, if you understand it, you're going to get this thing done in like 10 minutes at the most. And take a picture, turn it into Jupyter, and that is your assignment for assignment number 1. And that is all we got going on. So assignment number 1 is folding. And then after this assignment, you'll jump into DNA mutations. Don't forget, there's a video I made just like this for DNA mutations and goes over the 3 types of DNA mutations. But the reason we did folding first is because you won't understand what I'm talking about with mutations unless you understand what I'm talking about right now with how it folds to do what it's supposed to do. And then the 3rd assignment this week will be going over meiosis and doing a Venn diagram of meiosis versus mitosis. And that video is presented by one of our tutors. So make sure you watch that video. And then we have a quiz at the end of the week like we do every week. All right. If this doesn't make sense or any of the other stuff doesn't make sense, make sure you reach out to me, Jupyter message or text message. I will remind you that office hours are on Wednesday and Thursday from 2 to 2:30 through video conference. If you want to call or text me, you don't have to wait till that time. You can do it at any time. All right. Hopefully protein folding made sense to you. I wish you all good luck, and we'll see you on the flip side.